Okay, so I just want to first acknowledge the traditional custodians of the Great Barrier Reef and acknowledge the elders, past, current and emerging. So what I'm going to talk to you about today, well, first actually, I'm going to acknowledge the people that I've worked with on this, and the co-authors of this NESP review that we've created was David Westcott and Maria Byrne at University of Sydney, and Frederica Kroon, who's going to talk just after this. And then I also must acknowledge other staff at AIMS that I've had lots of discussions with about this, and they have done some editing of parts, and Peter Doherty edited a late version of the report. And also I want to acknowledge the NESP COTS working group, and that's been going on for several years, and it's been a very important forum for all our thinking and discussion in this space. So what I will present to you first is the aim of the project that we've done and the definitions, and then I'll go through the review so that I won't have time to go into the review in detail, but at least you'll know what sort of information is there and where you can access it. And then I'll just give you like a brief overview of the technologies that we have considered, just so you get a bit of a flavour for what we're talking about. And I'll show you the timeline estimates that we made for each of the technology options. And then I want to just touch on how this review is feeding straight into the new COTS Control Innovation Program that is led by the Foundation. Okay, so this was part of the same project that David talked to before, so Project 311, so implementation of the COTS research strategy. In there was aim to review developments in biological control and genetic technologies that can be used for pest management, because there is quite rapid technology developments in these fields, so we felt that it was time to actually go back and look at what's happening in this space. And the overall aim of this was to then provide the managers with up-to-date information on these options so that they can make decisions whether they want to support their development for COTS control. And also I want to say that it's very unlikely to find a silver bullet that's going to replace the manual control, but it's about looking for options that can complement the manual control. Like David was talking about these sites where they have to go back again and again and again, and they cannot get it below the threshold. So maybe there are some technologies that can assist in achieving that a lot faster. And then also we're looking for methods that could potentially reduce the frequency of outbreaks or the numbers in a logistically more efficient way. So just to take a step back, and a traditional definition of biological control, so the traditional biological control, is to reduce or eliminate a pest organism by interfering with their ecology, for example, by releasing predators or pathogens. Now, it's important to note that in this case, we're probably not aiming for elimination because it's a native species, so this is more for invasive species control. And also because we were considering a wider range of technologies, we went to so I just want to show you that there is a wider range, a wider definition these days for using biologically based control techniques. And if we adapt that very complicated definition to COTS, we can say that you can use either natural or modified whole organisms, or genes that they carry, or gene products, such as proteins that they produce, to reduce the effect of COTS, and then to favour desirable organisms such as coral. And in this report, we have reviewed a range of approaches, including traditional biological controls, use of predators and herbivores, and we included the herbivores because there are actually examples of release of herbivores into marine systems. So we wanted to include that in the review of what's happened historically. We also looked at microbial agents. And then we looked at the use of semiochemicals, which is communication molecules, and other push-pull strategies, and genetic biocontrol. So in this report, we went through all of these approaches and we presented the principles behind them, and then we went through the history of their use and their current use in terrestrial, freshwater and marine systems to see if there are lessons that we can build on there. And then we have a section where we actually go through whether these are suitable for use in cost control. So we present the relevant background, and in for some of these technologies, this is just about understanding the biology of COTS and the ecology of COTS as the background. In other areas, there has actually already been quite extensive research that's then reviewed in this report. Then we discuss how each of these technologies can be implemented as part of our integrated pest management strategy, and we discuss the possible risks and also critical knowledge gaps for each technology. And then we did a summary assessment where we actually scored each approach against 9 criteria relating to things like efficiency or expected efficiency, the risks, different types of risks, and also logistics. And with the logistics, we always benchmark against the manual control. And we also assessed, so we assessed both the level of knowledge for each of those criteria, and then we gave a score between 0 and 2, but obviously they were based on current knowledge, and those scores would be likely to change when more information come in. And then we gave some recommendations for the future. Also want to say that we have some more general sections in this report. So we discuss differences between terrestrial and aquatic systems. So you need to consider, if you want to take a technology that's developed for use in agriculture and you want to apply that in an open marine system, you need to take into consideration the properties of water that will influence both on the spread and dilution of your agent, and there is UV protection. Desiccation is a problem terrestrial environment, so there's a lot of different things to consider, plus also the more complex ecosystem and life histories of the organisms that are involved. We also discuss biosafety in there, and we have a separate section that go through recent developments in genetic technologies that are highly relevant, such as CRISPR-Cas9 for gene modifications and also gene drives. We discussed that as well. And then we have a section that touches on these other aspects, which is not our field of expertise. We highlight the importance of looking at modelling, environmental risk analysis, and partnerships with traditional owners, and obtaining social and institutional support, and the legislative context, and all of these things have to be done in parallel while you develop new approaches. So, what are these technologies? So if you use predators to control a pest, you have 2 different approaches. So you have the conservation approach, where you can use fisheries management to enhance the natural populations of predators, and Frederica is going to give a talk just after this that relates to that space. The alternative is to do an augmentative control, where you either relocate a predator of interest or you actually produce it in aquaculture and then release it. So the fisheries management can be applied on a broader scale, while the augmentative approach is more on a local scale. So you can say, if you have a reef that you are particularly interested in, you could put in an army of tritons in there. That's sort of the idea there. NESP has had a couple of other projects that relates to this space as well, and I'll just put it up there so you are aware. Semiochemicals. So animals produce molecules for communication, and this is actually quite prevalent in the marine environment in particular, because these chemicals can move in water. And the different strategies that we consider is attractants. So a COTS releases chemicals that can attract another COTS, basically. And you can imagine that perhaps you can use this to attract or to lure COTS out of hiding spaces. Perhaps this is a way that could be used on those really difficult reefs to get COTS that are hiding in crevices out when they are there to cull. You also have chemicals produced by predators, and I imagine most of you have seen this video where they expose the COTS to water that has had a triton in it, and it becomes completely erratic and just wants to get away. If you haven't seen it, just Google it and you'll find it. It's worth a look. Another approach is disturbance of spawning synchrony, and we have considered that as less likely to be successful in a marine system, and we discuss why. And this is more of a local application, we imagine. I don't think that's, you can't reach a GBR scale control using this, but you can use it on selected sites or selected reefs. So then traditional biological control with pathogens. So then we're talking about a control agent that self-replicates and can move across and cause disease, and there are viruses, bacteria, and protozoas. This is more complicated. Oh, that's actually, let's see. This is actually more complicated because with marine invertebrates, more and more there is reports that disease is actually polymicrobial. It's not one disease agent that causes disease. It's an opportunistic pathogen in an otherwise stressed individual. And also, a lot of the diseases that have been described are actually syndromes. So if you look at different locations and also different types of sea stars, for instance, for the sea star wasting disease, it's actually caused by different agents, and it's difficult to get a handle on it. It means it's difficult to find one agent that can be used for control. Another complicating factor is that if you use a virus that is the most likely to be specific, we actually don't have a cell line of COTS cells to produce them in. So this also really complicates this as an approach. But knowledge in this area can nevertheless be informing genetic control options. So if you understand how it causes disease in a COTS or how the immune response in a COTS responds to this, that's relevant for genetic control. And also when we looked at the microbiomes of COTS, we found this symbiont that might have potential as a specific genetic vector. And I'll come back to it. So genetic control strategies. So this is a very rapidly changing field with development of new technologies, but nothing is really known for COTS yet. So what we did was that we made a table of traits, so biologically and ecological traits that are relevant for development of genetic control systems in general, and then we looked at how the trait of COTS map onto that. And that excludes some approaches, while others are kept in. And then we looked at strategies that have already been used in other systems, and we also tried to think about possible strategies that are based on COTS-specific characteristics. So this is very much a theoretical exercise at this point, but we wanted to just have it there in one spot for further consideration. We also discussed possible ways of delivering this, because you can manipulate genes in the COTS itself, or you can have either free-living microorganisms or a symbiont that lives very closely with the COTS, where you manipulate them to either produce a protein like a toxin or a compound that's used in communication, or you can also get them to produce these interfering RNAs that can then interfere with the expression of specific genes in the COTS. So that's another way of delivering the modification. So we looked at the timeline estimates, and the manual control is already happening, obviously. When it comes to conservation approaches, we expect that they would be the first, the ones that would be ready first, because even though you need some time to put that in place, it's not involving a lot of development of technology. When it comes to semiochemicals, there has been a lot of research done already, but there are still some major gaps in terms of how do you actually deploy it and which exact compounds will you use. So there is still a significant lead time on that. The same with if you wanted to aquaculture produce tritons, there is still major knowledge gap in the production and also how to deploy it, and also all the safety testing that you would need to do. And with the genetic biocontrol, you can do some of these initial studies already, and then there would be even more needed to be done, including consultation and social license for their use. And gene drives at the moment not used anywhere in the world, not deemed safe enough, so pretty far off for use on the GBR, but the technology in this space is moving very fast, so we should still keep an eye on it. The report outlined the methodologies that are likely to be used, had a reality check on what the challenges are. We assessed the knowledge gaps, including the risks, and then we assessed the requirements for implementation. And we made some recommendations. Just want to say that now the CCIP is already starting, so it's feeding into that. And just as an example, this is just showing the proposals that were put forward from the population control area in the CCIP. And this involved a lot of the same people, but also some new people who's already worked in this space with different funding sources, and also some others who have relevant experience. And we based it on the analysis that existed in prioritizing which proposals that we put forward, and we were forced to do some prioritization of that. And this is under consideration as we speak, I think, pretty much. So we will know fairly soon if any of these proposals will be supported. Oops, and that's it.